Hello, today we will study about the histological structure of the stomach. As you know, stomach is the second part of the alimentary canal, which is more tubular than circular. It is highly elastic. Its mechanical and chemical action prepare the food for further digestion and absorption. Grossly, the human stomach is distinguished into three regions: cardiac portion with its dome-shaped swelling called fundus, the large carpus and narrow pylorus. These enclose a voluminous lumen. Histologically, the fundus and carpus are identical while the pylorus is markedly different one. So, similar to that of the esophagus, in the stomach, the wall of the stomach consists of four coats from inside to outside. These are mucosa, submucosa, muscularis externa, and serosa. So, we will study one by one. First of all, the outermost layer, first layer is nothing but the mucosa. When the stomach is empty, its inner surface shows many longitudinal folds called ruggi. When the stomach is filled with the food, the ruggi disappear. This is possible because of the loose nature of the mucosa. The surface is also divided by grooves into small, irregular, mammillated or gastric area. The entire surface epithelium is composed of very tall and equal basally nucleated mucus secreting cells which appear very pale when stained. The free surface of these cells form short microvilli. The epithelium is not a continuous layer but dips down to line millions of tiny gastric pits which are short in the fundus and carpus but long in pyloric region. The pits are basally connected to closely packed gastric glands. The surface cells continuously produce a viscous alkaline mucus which coats the stomach lining and protect it from abrasiveness of the food and from the digestive action of the gastric juice. Goblet cells are characteristically absent from the gastric mucosa. The lamina propria, because of the abundant, closely packed glands, contains poorly developed connective tissue strands. It also contains a few smooth muscle fibers, mast cells, fibrocytes, plasma cells, etc. Lymphatic nodules are scattered in the wall of the lamina propria. A thin but distinct muscularis mucosae lies in contact with the bases of the epithelial glands. It has two inner circular and outer longitudinal layers, but in certain places there is a third outer circular layer is also seen. Both lamina propria and muscularis mucosae follow the folds and valleys of the ruggi. Submucosa is a prominent dense connective tissue containing blood vessels, lymphatics, nerve fibers, mast cells, lymphoid wandering cells, and adipose tissues. No mucosal glands extend into it except in the juxtaduodenal area. The submucosal tissue projects thickly into the mucosal folds to form the core of the rugae. Muscularis externa consists of 3 layers instead of usual 2. The extra layer is of obliquely disposed fibers forming incomplete ring. It is innermost and circular muscle therefore form the middle layer. In the case of the stomach, serosa is the outermost coat. The glands of stomach is highly developed. They open into the bottom of the gastric pits but possess different types of cells. The glands are of 3 kinds: cardiac, gastric or principal, and pyloric. Of these, the gastric glands produce essential digestive factors of the gastric juice, while other two secrete mucus. The second cardiac gland occur in the cardiac part and are identical to the esophageal cardiac glands. They show a few argentine cells among the mucous cells. The next gastric or principal glands are the main secretory part of the stomach occurring throughout the fundus and corpus. They are simple or branched tubular glands which stain darkly and enclose a narrow lumen. They are mostly straight but near the muscularis mucosae becomes dilated and bent. Each gastric gland consists of 3 segments: the upper isthmus, which communicates with a fovea, the middle neck, and long and deep base. The isthmus consists of 2 types of cells: surface epithelial cells and parietal cells. The neck contains mucous neck cells and parietal cells, while the base is composed of chief cells, few parietal cells, and some argentine cells. Parietal cells, also known as oxyntic cells, are scattered in all segments of the gastric glands. They are large, acidophilic cells, rounded to pyramidal in shape, and possess prominent, centrally placed spherical nuclei. The cytoplasm consists of mitochondria. It also contains few rough endoplasmic reticulum and some free ribosomes. A Golgi apparatus is located between the nucleus and the cell base. These cells occur peripherally away from the glandular lumen and therefore communicate with the lumen by means of canaliculi, which occur between adjacent cells. These small passages are continuous with a network of intracellular secretory canaliculi formed by invaginations of the cell membrane. The intracellular canaliculi are lined with long microvilli. Parietal cells secrete free dilute hydrochloric acid into the intracellular canaliculi from where it passes to the lumen through the intracellular canaliculi. These cells also release a gastric intrinsic factor into the stomach. This factor is related to vitamin B12 metabolism. Chief cells, also called zymogenic or peptic cells, are the most numerous cuboidal basophilic cells bearing short irregular microvilli on their luminal surface. They show structural characters of protein synthesizing cells. Basal rough endoplasmic reticulum with abundant ribosomes, numerous mitochondria, and supranuclear Golgi complex and secretion granules which are large present in this region. These cells secrete the proenzyme pepsinogen. The argentaphin cells are also called chromaffin cells, are small, not very numerous. They are located between the basal lamina and chief cells. They contain many mitochondria, folded nucleus, and small characteristic basal granules which can be stained with silver or chromium salts. These are endocrine cells. Such cells also occur in the small intestine. All share a common property that all amine precursors uptake and decarboxylation and are therefore also known as apud cells. Some other cells or these cells secrete a hormone called gastrin. These hormones stimulate the oxyntic cells to produce their acid secretion. Some cells synthesize serotonin. This neurotransmitter brings about the contraction of smooth muscle fibers necessary for the peristalsis of the alimentary canal. The mucous neck cells are few and columnar. They show short microvilli and pale apical mucous granules. A large Golgi apparatus lies above the basal nucleus. The mucus produced by these cells is less viscous than produced by surface epithelial cells. Pylori glands are much shorter than the gastric glands. They are highly branched, tubular, and terminally coiled structure with a wide lumen which communicates with very deep fovilly termed pyloric pits. These are mucus secreting glands but occasionally show parietal and argentaphin cells. The distinguishing feature of the stomach are its wall is thick, ruggy is present, identical epithelial cells are seen, gastric pits are present, lamina propria packed with the gastric glands, parietal cells are seen, absence of goblet cells and villi occur, additional oblique layer is in muscularis externa is seen. So these are the distinguishing feature of the stomach.